Thank you. I wanted to tell you today very briefly about strategies that we are employing at Stanford, funded by CIRM, for improving tissue healing and regeneration through the activation of endogenous stem cells. And this work is predicated on this slide going forward, on the fact that stem cells are key regenerative players in tissue healing. They're activated by injury, and in response to that, they control whether tissues heal or the extent to which they can heal after injury. I'm sorry, but it's not advancing. Oh, there we go. Thank you. So a core concept behind all of our work is that we use the Wnt protein as a therapeutic. Wnt proteins activate stem cells, and the extent to which they get activated controls whether the tissue heals or not. We have as our target population individuals over age 50. This is because Wnt signaling declines with age, and with that decline is a decline or a shift in stem cell behavior. Stem cells are slower to respond, and in doing so, tissue healing is slower as well. Wnt signals are important for the maintenance of bone, both its homeostasis and its healing. So you can see on the top the skeleton, the bone mass of a young individual and that of an aged individual. We consider this our therapeutic option, and it represents large opportunities, we believe, for advancing a Wnt therapy. Sorry. So our basic strategy is this, that we seek to activate stem cells using a Wnt protein therapeutic. It's in a proprietary formulation, and then these activated stem cells then contribute to bone repair and regeneration. We believe that this has a broad potential to apply to a number of degenerative conditions. So as I mentioned, Wnt proteins activate stem cells. It controls their self-renewal and their proliferation of embryonic stem cells, adult stem cells, and allogeneic stem cells. Here's an example of that using stem cells from aged individuals. These are animals, I should say. And you can see that the protein, single exposure to the protein leads to strong activation of Wnt signaling in all different kinds of stem cell populations. This is in response to a single dose of the liposomal formulation of Wnt. This is a quantitative reverse transcriptase PCR reaction. So our ex vivo strategy is this. We harvest an autograft. The autograft is the standard of care for bone defects. This autograft is, in this case, coming from an aged animal. The autograft is placed on the back table, and during that period of time, we ask that the autograft get treated with a liposomal formulation of Wnt. And here you see what typically happens to an autograft on the back table, that the cells start to die. And in this picture, they're labeled with a protein that recognizes fragmented DNA. So in a side-by-side comparison, an autograft treated with a liposomal Wnt shows significantly reduced cell death. Simultaneously with this effect, we see an increase, transient increase, in the level of cell proliferation. And here that's visualized using the incorporation of a thymidine analog called BRDU. So this transient increase in cell proliferation and a significant reduction in cell death leads to superior engraftment of the autograft. Simultaneously with this, we monitored the expression of anabolic osteogenic genes. And here you can see that all of them, RUNX2, ostrix, and osteocalcin, all osteogenic markers, are increased within a very short period of exposure, a single exposure to liposomal Wnt. So back to the application. Following the Wnt treatment, the autograft is briefly rinsed using a coffee filter-like device, so no special equipment needed, and reintroduced into the bone defect. And here I show you one example of disease-modifying activity in spine fusion. On the top panel, you see an autograft treated with PBS and an autograft treated with the L-Wnt3A. And the micro-CT, it's highlighted in pink. So you can see that the size of the autograft is equivalent at the time of surgery. But by 49 days, you can see that autografts from older patients are not very osteogenic, and so there's a lack of bone. Whereas in the single exposure to a Wnt, you see significant bone formation, and that's shown here. And they go on to show superior healing in a shorter period of time. In addition to bone, there are a number of other therapeutic applications that we consider viable strategies moving forward, and they include articular cartilage, hair, and skin, all because the stem cells are responsive to a Wnt stimulus. Our funding stream has been through CIRM, and we're grateful for their support because they support us in the earliest stages of our translational award through bridging and extraordinary supplements. And we're excited about the fact that we're now partnering with Ancasa, an Avalon-backed company, and we're moving now forward into towards IND filing. We anticipate being there in about 30 months. In addition, there's a CIRM grant that allows a partnership between companies and institutions, and this is called a Preclinical Development Award. And if we're fortunate enough to receive that grant, our goals will be to continue with CMC process development, early safety talks, and some assay development. So I want to also point out that we have ongoing studies in these areas of hair, skin, and articular cartilage. So I want to thank the people that made this work possible and the institutions that have supported us as we go forward. Thank you very much. Yes. Yeah, so that's a very good question because Wnt, as well as being activators of stem cells, they also activate cancer cells. And so that's been a key concern of ours, especially given that other growth factors on the market that address this niche have been associated with black box warnings from the FDA because of cancer risk. So that's why we developed an ex vivo strategy. So the Wnt protein is never introduced into the body for exactly the reasons that you mentioned. It's also not delivered systemically, and this is important for safety reasons. So the ex vivo strategy limits the exposure to the duration and the time of the application. Then it's rinsed to remove any free Wnt, then reintroduced. Yeah. Yes. So he asked, what is the duration of the ex vivo treatment? We find that within a 1 hour, 60-minute exposure is sufficient to activate the pathway. So it's significantly elevated. We can increase the exposure up to 4 hours without loss of activity and still see the effects of preventing cell death and enhancing proliferation. So we think a minimum exposure, well, it might be lower. We haven't tested lower, but we think 60 minutes. Yes?
So is the size of autograft where we are also determining the duration? So I will give how much penetration?
Yeah. So we asked, is the size of the autograft a limiting factor because of the penetration? So it turns out that it's not. The liposomal formulation of the Wnt penetrates rapidly, and it reaches the inside just as soon as just through diffusion. It's because the autograft is also not a solid body. The tissues are activated at the same time, both in the center as on the periphery. So size is not a problem in this case. Thank you very much.